In this video, I'm going to talk about some of the chemical properties of amino acids, which are the constituents of protein. And so there are 20 amino acids. So I'll just call it AA. And these, well, there's more than 20, but 20 are the ones that we are interested in for humans. And these are the constituents of protein. So a protein is just a polymer, or as we call in chemistry and biochemistry, a polypeptide of amino acids. And so to talk about proteins, we first need to talk about their constituents, which are these amino acids. And right here I have this chart of amino acids, and this has it broken down into different chemical properties. And so up here in this top area, we have the electrically charged amino acids, and those are things like arginine, histidine, and lysine, which are the positively charged amino acids. And this charge, by the way, is at physiological pH, which is pH around 7.35. So our body, at least in the blood, regulates our pH very close to this. It's usually in this range, and so these will be positively charged when it's at this physiological pH. This primary amine on the lysine, and this guanidium group here at the end of the arginine. This will be positively charged. The histidine, it's hard to see right here, but this does have the pKa, which is around 6.0. And so this is actually going to be in a range where it's going to be mostly in this deprotonated form, because when we're at pH 7.35, then a pKa of 6 is going to let go of an extra hydrogen. So if this had an extra hydrogen on here, it would be positively charged. But since this has a this fairly low pKa, it's going to be somewhere in the neighborhood of probably, I don't know, maybe 70-ish% will be in the non-charged form, and 30-ish%. Those are just percentages I'm sort of ballparking here. So 70% would probably be non-charged and 30% charged. But we will also see in this video and in the one on proteins that you can control the environment that an amino acid is in the protein, which can adjust the pKa. So this is just the pKa of, the pKa of histidine, sort of just in a solution of water. Then we have the negatively charged amino acids here. They're still charged, but they are negatively charged at physiological pH because they have these carboxyl groups on the end, which are in weak acid. If you remember from before, they have something like this, and then if it's in the protonated form, it'll have that H there. But you can, I don't know if you can see in your screen, but these have a pKa. This one says 3.71. This says 4.15, I think. So those are those pKa's are getting a little bit lower, so this hydrogen will most likely be off, and this gives them this negative electric charge. Down here we have the polar but uncharged amino acids, and you can see the side chain on serine, for instance, has this hydroxyl group. Threonine also has a hydroxyl group, but then it has this uh, CH3 group coming off. Uh, Asparagine and glutamine are have these right here. So instead of a carboxyl group, it has the oxygen that goes down to this nitrogen here, and so this nitrogen is not as is not going to pick up hydrogens as much because its lone pair is in a resonance structure with that oxygen there, and so that stabilizes the nitrogen in the neutral form. And so this is neutral, but there is still hydrogen bonding. This oxygen here can act as a hydrogen bond acceptor, and this nitrogen with the two hydrogens on it, the hydrogens can act as hydrogen bond donors. Then we have over here, it calls it the special cases. And so cysteine is one. It has this sulfur, so it's the same shape, it's the same geometry as the serine, but instead of an oxygen, it has a sulfur. And we'll talk about later that having a sulfur there reduces the pKa. It stabilizes. The sulfur is more stable when it's charged than an oxygen. So that pKa is, I think that says 8.14. And there are other things that you can do with this cysteine, as we'll talk about later. Selenocysteine. So this is something that happened. This is a amino acid that's, as far as I know, it's exclusive to bacteria. Well, I don't know if it's just bacteria. It might be in archaea as well. I don't know that much about archaea, but it is. We do not find this in humans. And so this is the one that I'm not really going to talk about. So I said there's 20. This says it has the 21 amino acids. So this is that 21st. Then the glycine is different because it doesn't have any side chains. Well, for one thing, this bond right here is very free. So glycine is often to use a very scientific word, it's more wiggly, and as we'll see later, here in a little bit, it makes it non-chiral or achiral, so that makes it kind of a special case. And then proline is another special case because it forms this ring that, so, I mean, there are other amino acids that have rings. We saw one here, but this ring goes from its alpha carbon here to its nitrogen. And so this constrains some of the bond angles that we have in here, and we will talk about that more later. And so down here we have the hydrophobic amino acids. So these are the ones that, while they're hydrophobic, they don't mix with water very well. This just shows it in sort of increasing side chain size. We see over here at this end, we have side chains that have these double bond ring structures going on. So those are called aromatic. When you have this alternating double bond going on in the ring structure here, so those have this aromaticity, which we will discuss some of the properties of that more later. These ones just have these differently branched hydrocarbon chains. Methionine has a sulfur in it, which makes it a little bit different. We're not really going to talk that much about this sulfur. It doesn't figure into to what we're going to discuss here that much. So we talk a little bit about stereochemistry in the carbohydrates video, and I talked about the difference between L, the levorotatory, and the D, which was the dextro dextrorotatory. There we go. And I said that this just has to do with how it shifts the angle of plane polarized light. So if we had light that was polarized completely upward like that, if we sent it through a mixture that had equal parts L and D, this would not change. But if we sent it through one that had more L than D, or if it was just all L, then it would maybe shift this way by some degrees, by some theta degrees. And same as if we sent it through one that was mostly this or all this, it would shift the same degrees but in the opposite direction. And the angle that it shifts by tells you something about the relative concentrations of these two. And so if we had just a little bit more of this than this, then maybe it would only shift slightly. But if we had a lot more of this than this, then it might shift more. And so that's just a little bit more about this levorotatory and dextrorotatory. I said in the carbohydrates video that carbohydrates, that sugars are almost always, at least as far as I know, they're always in the D form. For amino acids, it's the opposite. They are always in the L form. So all the amino acids that we use are in the L form. Once again, I know bacteria have a few small but notable exceptions of using D amino acids for very specific purposes. One of them is they use them in their cell walls. Those are the amino acids. It's those amino acids in the cell walls that our antibacterial or antibiotics target. I'm not going to go too far into that because that's a different subject. Down here, so I talked about in the carbohydrates video as well that there's a difference between the LD notation and the RS notation. And so there are 19 chiral amino acids. Like I said, the glycine is achiral since it has two hydrogens bound to its alpha carbon. And so there isn't a mirror image of that, or I mean, you could have a mirror image of it, but then you could rotate it around in space such that you could superimpose it onto it itself again. And so 18 of the 19 amino acids have the S chirality, and the one exception is cysteine, the one with the sulfur. I mean, the sulfur doesn't have to do with anything to do with its it being an R chiral, but it just makes it easier to remember that that's the exception to the S rule. That cysteine is the weirdo. It's the one with the R chirality. It is still L. It still has the levorotatory effect on plain polarized light, but it is the L chirality. And so yeah, like I said in the carbohydrates video, the LD is talking about the entire molecule, where the RS is talking about just a single carbon. So you can have you could have two chiral carbons in your molecule, and one of them might be R and the other S, but the whole molecule would be either L or D. And so that's kind of the difference between those. Um, so down here, there's just a bit more about the acid-base chemistry of amino acids, and so a few of them are listed here. We see that that serine has a pKa of about 13, so the hydrogen on here is not very acidic. If you remember, the lower the pKa, the more acidic it is. Threonine is about the same, about 13 pKa. Once we get to tyrosine, even though we still have just an OH, it becomes more acidic, and that is because, as I show here, when you have oxygen bound to a ring structure like this ring structure gives it this resonance stabilization. So resonance stabilized, and so that charge can be sort of shared. See it right there. There. It can be shared throughout this ring, and so that helps stabilize the conjugate base and makes this more acidic. I have down here, you can see just the difference between a aromatic ring and a non-aromatic ring. The pKa in the aromatic ring, which when it has a hydroxyl on it, is called a phenol, and the cyclohexanol, so it's 6-hex, cyclo, it's a circle, and the OL just tells you that there's an alcohol or hydroxyl group on it. And the phenol has pKa of 10, the cyclohexanol has pKa of 16, so that has a fairly large impact on the pKa. And then if we look at the cysteine, and we see that this is quite a bit more acidic than the serine, even though they are geometrically the same. The only difference between this has sulfur and this has oxygen. And this has to do with the size of the atoms. So the oxygen, which if you remember on the periodic table is in the same column as sulfur, but it's one above it, and so it has one less electron shell than the sulfur. And so what happens is when you have this larger electron cloud, it can accommodate this this negative charge better, and so that allows it to be in the negatively charged form more stably. If is stably a word? It is now. So anyway, that's why the serine, the hydroxyl, the alcohol group on serine is less acidic than the sulfur group on cysteine. Over here, I have a little bit more about some of the acids and bases on amino acids. So the terminal carboxyl group, which would be like this one right here, not the side chain one, but the one on the alpha carbon, that has a typical pKa of around 3.1. The amino group, which would be like this one here, has typically around 8.0. And so at physiological pH of 7.35. This is most likely going to be in the protonated form, and since this is so low, it's most likely to be in this deprotonated form, so negatively charged and positively charged. And then, once again, we just have the aspartic and glutamic acid, which had the carboxylic acid on it. The histidine we talked about above, which has the pKa around 6. The cysteine around 8.3. The tyrosine, this is at 10.9, this is at 10.1. So yeah, that's kind of a good place to go. So I have this sort of double boxed here, and it says that the pKa values depend on temperature, ionic strength, and the microenvironment of the ionizable group. And that's why a lot of times you will see pKa's from different amino acids or even other acids being slightly different from each other. If we're, this has the 10.1 and this has the 10.9, and that has to do with the environment of the molecule. So ionic strength is just the amount of different salts that you have in your solution. So if we say had acetic acid and we were in solution and we started adding a bunch of table salt, NaCl, which dissociates in solution into Na plus Cl minus. And so you see if we have 0 of it, then we are at that 4.7-ish range, of pKa of around 4.7. But then as we start adding this salt and the pKa starts dropping, and so we start losing this hydrogen more readily because the pKa is dropping. And the reason for that is in the conjugate base form, after it's deprotonated, we now have these sodium ions around that can help stabilize this negative charge. And so that's why it's going to more readily become a charged molecule, because the charge can be stabilized. Then over here we have pKa versus temperature as we increase the temperature. So these are, so this one up here is the imidazole, which looks like this. So it's talking about this hydrogen here. And so we see as we increase the temperature, the pKa is dropping for the imidazole. Then down here, but is still just sort of following the same path, we have the histidine, which you see has this imidazole group on the side chain of it. And the reason why it would have a lower pKa than the free imidazole has to do with the fact that it has this other stuff here bound to it. And so these are affecting the electron density throughout the entire molecule in different ways, and that will change the way that this right here behaves. And so that's why we get the lower pKa's, even though you can see that it's following the same trajectory down. Then the green here, which slightly goes up, the pKa slightly goes up, that's the citric acid. So we see this has 3 carboxylic acids on it, and that pKa is going to go slightly up as we increase temperature. Down here we see in the purple here, both of these stay pretty even. The purple one is the glutamic acid, so the pKa of this doesn't change that much with increased temperature. The oxalic acid, which is this sort of double acetic acid together. Uh, the temperature doesn't affect the pKa of that as much. But we can see there's the temperature has a bit more of a dramatic effect on some of these other ones. So the ionic strength, or basically the salt content of your solution and the temperature of your solution, are going to change the pKa. The environment is also going to do that. And so I have here the pKa's of different acids in different solvents. So here we have it in water. We can see phenol, which once again is about 10 pKa of 10, which we saw before with the tyrosine. Acetic acid about 4.76 in water. Hydrochloric acid, that says minus 5.9. There's a little A there because there was some footnotes on here for sources, but I didn't include that in here. So it's around -6, which we've seen before. The sulfuric acid, the first hydrogen on it is about -9. But then if we put it in a different solution, so say we put it in this acetonitrile solution, which you can see is going to be different than water, which is a very polar molecule. So this will have some polarity to this triple bond nitrogen, but it also has this methyl group here. And so what we see is our phenol goes from pKa of 10 to pKa of almost 30. And so when it's in something that is more hydrophobic, it's not going to want to lose its hydrogen as readily, because in a hydrophobic environment the charge is going to be pretty much effectively stronger. And that is important, as we will see in the next video when we talk about protein structure, that having a charge, so if we have something like an oxygen that's negatively charged and it's in a hydrophobic environment, there's nothing stabilizing that charge. And so it's not going to want to give up its hydrogen nearly as readily. And we can see the same thing with acetic acid, which goes from a pKa of 4.76 to 23.51, so almost 24. Hydrochloric acid, which is almost -6 pKa in water, goes up to 10.3. Sulfuric acid -9 goes up to almost +9. And then if we are in dichloroethane, which is this molecule right here, we see that we go up in our pKa, but not as dramatically as was in the acetonitrile. Then GP is just in gas phase, so that's the pKa when it's evaporated. And you can see that the pKa goes up insanely far in the gas phase, so from 10 to 251, from 4.76 to 250, so they're all around 220 to 250. So when they are in the gas phase, they do not have they're not very acidic. But we see if we go into these hydrophobic solvents, then our pKa's increase considerably. All right, going back here, so still looking at this acid-base idea. I just have here this idea that at a very high pH solution, so high pH, we're going to be in this maximally protonated form, so it's going to have a net charge of +1. As we increase the pH, or yeah, this should be low pH, sorry. So that should be low pH. So lots of hydrogen ions in solution. And so as we increase the pH, you will see that we start deprotonating things, and this will be deprotonated first. Then this will be deprotonated. Now we have a net charge of -1, and then eventually we will deprotonate the side chain. And so right here, once again, we're just looking at the imidazole from histidine, and we see that it has a pKa of a 6, and when it's protonated, it has this resonance here. So this lone pair will collapse down on there, collapse up in there, and so it's resonance stabilized, and so that's why it will protonate. In tryptophan, which has this larger ring system, the electron lone pair in the nitrogen is actually involved in the aromaticity of the ring. And so this ring is aromatic all the way around. And that's why this nitrogen here will not pick up an extra hydrogen. So I have pKa equals question mark here because there is not a pKa for this nitrogen. And you can see how this the resonance of this involves the lone pair on this nitrogen in the aromaticity of the tryptophan side chain. Over here I just have a titration curve. So if we keep adding these groups here, which are negatively charged, then we will be pulling off the hydrogen on our carboxylic acid groups, and so our pH will slowly increase, and we get into this area here, which is the buffer zone, and this is very common for weaker acids, things like carboxylic acids, that they have this buffer zone. We're adding, so if you add from here to here this amount of OH minus, you are increasing the pH by quite a bit, whereas over here if you add the same amount, your pH is not increasing by that much, and so this is a buffered area. And here we have the pH equals pKa, and we talked about in the acid base videos that the pH equals the pKa plus the logarithm of the conjugate base over the acid. This was the Henderson-Hasselbalch equation. And so when this and this are equal to each other, then this is 1, and logarithm of 1 is 0. And so pH equals pKa in the center here, when we have just as much of we have 50% of our protonated form and 50% of our deprotonated form. All right, let's move on a little bit from this acid-base chemistry, and so we will look here at these. So these are called the beta branched amino acids, and what that's talking about is we have our alpha carbon on these amino acids right here, which is the one that has the carboxylic acid coming off, the amine coming off, and the side chain coming off. And then going in the Greek letter order, we go to our beta carbon on each of these. And you see that on these 3 amino acids, the beta carbon is branched in that it goes to two different species that are not just a hydrogen. they go to bulkier species. And so these beta-branched amino acids are constrained in how much you can rotate around this bond here, this alpha to beta bond, and that's because when the amino acid is incorporated into a protein, then because these large bulky groups are very close to what will be the backbone of your protein, there can be more steric interference, more clashing going on with it. And so this down here just kind of explains that what ends up happening is because of this. And we'll talk about these secondary structures more in the next video. You will not see these in alpha helical secondary structures that often because of this beta-branched structure here. And I have this down here because if you are into nutrition and things like that, you've probably heard of the branched chain amino acids, or BCAA. So you can buy supplements of branched chain amino acids. These are different from the beta-branched. You see that isoleucine and valine are both beta-branched and branched chain, but leucine, because its beta carbon right here is not the one that's branched. It's branched further up here. It is not beta branched, but it is still a branched chain amino acid. And in later videos, we will talk more about the nutritional side of these branched chain amino acids and their role in stimulating muscle growth. But I'm not going to get too much into that just yet. I just wanted to point out that the beta branched and the branched chain amino acids, even though there's a lot of overlap, are not the same thing. Alright, and so now let's talk a little bit about cysteine and what makes cysteine one of the special cases of amino acids. So I also have methionine drawn here because it also contains a sulfur, but it's the sulfur on cysteine that we're going to be concerned with here. What we're talking about here does not really apply to methionine, but I just wanted to show that the structural differences between these. And so with cysteine, what can happen is you can actually do redox reactions with this sulfur, and through that you can form what are called disulfide bonds, right, like what you see here. And so by oxidizing the sulfur, so this should actually probably have a 2 here because we're forming it with 2 different sulfurs, and if this was in a protein, this wouldn't be an H, but we would say that's an R that goes to another cysteine. And so if we oxidize that, we remove an H plus, so it's removing an H plus, so we can say plus 2 H pluses, and we're also removing 2 electrons, so 2 E minus, which is how chemists, well, I think physicists do too, represent electrons. And so that should have a 2 here. So we oxidized. Oxidation is loss of electrons, so we've lost these 2 electrons, and we've also lost two of the hydrogens. And so that's how we've oxidized this to form this disulfide bond. Then it can be reduced again to reform our our normal side chain here. Uh, this is getting kind of into protein structure, but I just wanted to show an instance of this happening. So this this is the insulin molecule, which is composed of two polypeptides. So these are two different polypeptides. And these yellow, see from cysteine here to cysteine here, are disulfide bonds forming within this chain. And then we have two of them forming between the two chains. And we see the structure down here. We can see the yellow right there, and right there, and right there, representing the disulfide bonds between the two differently colored chains here. And so insulin is a good instance, a good example of these disulfide bonds being used in holding a protein together like this. Another place where you see those disulfide bonds are in glutathione, which, once again, if you are into health type things, you may have heard of this molecule. It's an antioxidant. It's sort of our natural antioxidant molecule. And we see this is made of a glutamate, a cysteine, and a glycine. It's different than a peptide because on this we are bonding to the side chain of our glutamate rather than to one of these groups over here near its alpha carbon. So this molecule isn't really a peptide in the sense that we would talk about in proteins. It's sort of a special molecule, but it does contain this sulfur here, which we can see up over here, and we can, if we oxidize it, so that's why it's showing oxygen coming in, that's stealing the hydrogens and the electrons from this, and we form, so this has a 2 here because there's supposed to be 2 of these, is forming this disulfide bridge here. And doing this reaction is actually how it performs its antioxidant function. By oxidizing this sulfur to make a disulfide bond, it's taking the electrons from this oxidative species, and so that this shows it as oxygen, but it could be other reactive oxygen species, and so it's oxidizing itself and thereby reducing the reactive oxygen species. Over here, we're going to talk a little bit more about these aromatic rings and why those are special and interesting. And aromatic just is one of those ring systems that have the alternating double bonds. And what ends up happening with these is it makes them interact with light differently. All of the other amino acids will absorb light that is much higher frequency, so it's much further into the UV range. These ones have this absorption spectrum here in the lower frequency UV range. It's getting somewhat close to visible light range, and so the tryptophan, the tyrosine, the phenylalanine, these are often used. These absorption maxima are often used when you were using UV-Vis spectroscopy to study proteins spectroscopy. And histidine is in here too. We see on here that tryptophan, which was the one that had that, which has this double ring species. So it's very aromatic here. Has this very high peak, so that's often the one that you're looking for in UV-Vis spectroscopy. And so as far as this aromaticity, which is a special type of what's called a conjugated double bond. And so that's a special case of the conjugated double bond, which just means alternating double bonds. So you might have a chain like this, and then every other bond is a double bond. And so over here we have these rules about how things affect the maximum peak in our UV-Vis spectroscopy. And so here we see that if we have alternating double bonds like this in different rings, then our base value, our base frequency or wavelength rather, where we will have our maximum peak is at around 214 nanometers, which would be somewhere not even on this graph. It'd be somewhere over here. If our base value has the double bonds within the same ring, then it jumps up to 253, which would be somewhere like around here. And then we see every time we add another double bond, we add plus 30 onto onto the wavelengths, or where the maximum peak is in wavelengths. And so we see if we have something like this benzene group, which is all double bonds in the same ring, and we have three of them. So in the same ring, we're at 253. Then we add 30 more because we have three of them. So we should be at around 280. Well, look, that's exactly where we find this peak here. And there are other things that affect where the maximum peak is. Things like alkyl substitutions, which are just hydrocarbon chains. So it could be a methyl group, it could be an ethyl group. So things like that. Exocyclic double bond. So that would just be, so if we had like another double bond here, but then we also had this exocyclic double bond. Acetate group would be something like this. Ether group is something like that. Thioether, something like that. Bromine and chlorine would just be adding one of those onto it. Secondary amine group would be something like that. And so we see that those all can add to the wavelength of our maximum peak, where on the bottom here our maximum peak would show up. And we have an example here too, where we have what's called the Dihedral annular ring, which just means that the double bonds are in separate rings, which means we start with the 214. It has 4 alkyl substituents on it. So that would be things like that. So that's 1, 2, 3, 4 alkyl substituents. So alkyl is plus 5 times 4, we get 20. Exocyclic double bond is just because these are in 2 different rings, so that's another plus 5, so 239. And so we could expect to find a peak, our maximum peak, at 239 nanometers. And the reason for this is when we're adding these double bonds, so we see here molecule with only single bonds, the energy difference between uh, a electron in this state here and then an electron in the excited state up here is very large. So that's higher energy, higher frequency, shorter wavelength. And so the wavelength is going to be somewhere further off in the UV here. Then if we start adding double bonds, we start closing this gap in energy. So less energy, lower frequency, longer wavelengths. Then when we have these conjugated double bonds, there's very little difference between the electron in its ground state and the electron in its excited state, such that when it falls back down, it releases energy as light. And so if you get a lot of conjugated double bonds, you will actually start seeing the molecule in invisible color. In fact, as we'll see in later videos, things like vitamin A, which I don't remember the structure offhand, but it has some things like this, but it also contains just a bunch of double bonds, and that's why it will appear like it's orange rather than if you just have like some amino acids as a powder. If you just buy some powdered amino acids, they'll probably look white because they don't have enough conjugation to get out of to drop out of the UV range into the visible range. But as you keep adding more and more conjugation, then you will start seeing things in the visible range. And so that's why if you look at the molecules for things like different colored dyes, they will oftentimes have lots of double bond conjugation. So another interesting thing that we see with these aromatic rings is because the double bonds are made from these pi orbitals, these p orbitals, which we can see are delocalized like this. We can have this what's called pi stacking. So pi stacking. And what that is the p orbitals on these will overlap with the p orbitals on another aromatic ring like this. So that one will be there, and this will be there, and those will be overlapping, and these will be overlapping. And so we can get overlapping here, and that stabilizes the position of these, and we get this pi stacking, which causes these to sort of stay in this position. They have sort of a weak bonding like nature to them, and so we see that quite often in protein structures. It also occurs in DNA because the bases in DNA have some aromatic nature to them, and so we see pi stacking with that as well. So amino acids have these things called dihedral angles. Well, first I guess I should talk about the bonding of two amino acids. So we form a bond between the nitrogen of one and the carboxylic acid of the other. This shows these blue here come off to become water while these attach to each other. So what happens is the lone pair on this nitrogen will attack that partially positive carbon there, which will kick the electrons up to there. We get an intermediate which will it'll have something like this. So this should be bound to that. and this will go on to the rest of the amino acid. This will go on to the rest of its amino acid. We'll call them R1 and R2. This has hydrogens on it. This will still have its OH on it. This OH here. Then what happens is the lone pair of electrons in this will collapse back down. Carbons only ever want 4 bonds, so something's got to go, and this will probably be it. So this bond here will kind of reach over and grab that hydrogen there. So those electrons will go back to the nitrogen, and then we end up with our R1 going to C double bond oxygen from up there. This goes to the nitrogen, which is like that, and this right here is now the OH2 or H2O, and so we lose that water to come off here and form this peptide bond here. And so this is called a peptide bond, and this nitrogen would have a lone pair on it, and this peptide bond is stable, or its geometry is stable, because what we have is should probably have written the lone pair here and the hydrogen there. I'll redraw it right here. So we have our carbon like that. This goes down to the nitrogen, which has the hydrogen here, and then this goes up to that. And so this lone pair can go here, kick those electrons up there, and so we have R1C up to here, negative, double bond down to nitrogen, which is positive now, and up to there. And so these are resonance structures of each other. These are resonance stabilized. And so we see that there's a slight double bond character here, which prevents this from rotating around. We cannot rotate around that bond, and so what we have are these planes on here. And so we can think of this as kind of like having this plane that's always flat on here. And that means we end up with these what are called dihedral angles. See, this is that plane that I was talking about. So dihedral angles, and it's basically just the angles at which these planes sort of flap around the alpha carbon on our amino acid. And so we have the psi or psi, I would just call it psi, the psi angle and the phi angle here. And so the phi angle is the one that points towards the nitrogen on the amino acid, and the psi angle is the one that points towards the carbon double bond to oxygen. And these phi and psi angles, because of steric hindrance with things like the side chain on this amino acid and the other amino acids, have only these allowed angles in which they can exist. And so this is what's called a Ramachandran plot, and it's just plotting the two angles. So down here we have the phi angle from negative 180 to 180, and if you know your geometry, if you go if we're at a circle and we're here, if you go negative 180 degrees, you will end up in the same place as if you go positive 180 degrees. And so this is actually circular. Uh, this point right here is the same as this point right here, and same as when we're looking at the psi angles going up here. So it goes from positive to negative 180. But we see here that we can only remain in this range in the phi psi angles, or we can be down here. There's a small allowed region over here. And so this constrains the angles at which these amino acids or these peptide bonds can rotate around these single bonds going to the alpha carbon, and that affects the folding of protein. And over here, so proline is a little bit different than a lot of them, because we see here our phi angle is massively constrained by the fact that we have this ring structure here, and so we don't get any kind of rotation around this bond here. So this right here is the psi angle. We can still move a little bit as long as we're in these allowed ranges here on the psi angle, but the phi angle is constrained by the fact that proline has this ring here. And so proline has some of its own properties as far as how it behaves in a protein. We'll talk about that more in the next video. It's known as an alpha helix killer. So if we find this in a protein, and it's at if it's in an alpha helix, it'll be at one of the ends of the alpha helix, because the way that this geometry works, it's not conducive to alpha helix secondary structure, which, like I said, I'll talk about more in the next video. One more thing I want to cover for amino acids is some of the modifications we can make to them. So the most important one that we need to know about is phosphorylation, and that's just adding a phosphate group. Phosphorylation, that is adding a phosphate group, and this is pretty much always on serine, threonine, or tyrosine. So the serine, threonine so these are often kind of grouped together. We call them serine threonine kinases. A kinase being a type of enzyme that phosphorylates these amino acid residues, these side chains, and so we usually group the serine threonine kinases together, and then we will also have the tyrosine. That should be tyrosine kinases, which are usually different from the serine threonine kinases. So we see here we have our tyrosine with a phosphate on it, our threonine, and our serine with a phosphate on it. And this is just the mechanism by which that happens. So in a protein, we will often have some kind of base here that will deprotonate this the serine here, or it could be the same thing on a threonine or a tyrosine. Then that will attack the third phosphate on ATP, and then it will take the phosphate off of that, turning it into adenosine diphosphate, ADP, and thereby phosphorylating our hydroxyl group, our alcohol group on the on the protein. So these little squiggles mean that there could be a bunch of protein going off from that, and so this is something we will run into all the time in later videos. This phosphorylation, because this is a way that protein function is regulated. So the adding and the removing of phosphates. Usually when you remove it, it doesn't go back onto an ATP. It'll just sort of go off into solution. But adding and removing phosphates is the major way that proteins in our body are regulated. So some proteins are shut off when you add a phosphate to them. Some of them are activated when you add a phosphate to them. Some of them, and the reverse can be true as well. So this is a major way that proteins are regulated. And so we have our serine threonine kinases, which are enzymes that add phosphates. So kinase add phosphates. Phosphatases are usually ones that remove phosphates. So kinases add phosphates, add PO4, and these remove PO4. And the same thing with tyrosine. We can have phosphatases for that, phosphatase. And so, yeah, that, like I said, that's a major way that proteins are regulated, by phosphorylating and dephosphorylating these serine, threonine, or tyrosine amino acids that are incorporated in the protein. Another thing we will see, so this is just two instances of what's called glycosylation, and that is adding sugar to amino acids in your protein. So this is what's called glucose. Well, I don't remember exactly what. So we usually just call it glicnac and galnac. But anyway, these are sugars that are added to our protein here, and so they can be added to asparagine or serine, and you can add a bunch of different kinds of sugars. These are just one example. So you can add a bunch of sugars. You often see it where you have giant sugar chains. They might even be branching, and this will be attached to our protein. And so you can have multiple sorts of chain branching coming off of here. And so this is glycosylation, and this is not uncommon in proteins. And so we can have these different types of glycosylations to serines and asparagines and things like that. So down here I have acetylation. We will not run into this that often in what we are going to talk about with metabolism and nutrition, but this happens on histones, which are proteins involved in nucleosomes, which are bound to DNA, and so this acetylation and also methylation are things that happen to usually to lysine. This is a lysine amino acid. These squiggly lines meaning that it's part of a protein. And so we can acetylate, which as this acetyl group on here. You can also add methyl groups on it, so that would be our NH, and this is going over to the rest of the lysine, and then we'll have CH3 groups on here. So things like that. And so that these are on histones, and I'll talk about histones a bit more in the in a video in the future when I talk about DNA. But this is, if you've heard of things, if you've heard of epigenetics, that's what this is. So histone acetylation and methylation controls the expression of genes, and so this is another sort of modification that you can see on amino acids. Like I said, we're not going to talk that much about it in this playlist, just because it's not as relevant to nutrition and metabolism, but I just wanted to make you aware that this is another kind of amino acid modification that you might run across. And so I think that was everything I wanted to talk about. So yeah, we talked about the different properties of amino acids. They can be positively or negatively charged at physiological pH. They can be polar but uncharged. We have these special cases, things like the cysteine, the glycine, and the proline. We can have the hydrophobic, the uncharged, unpolar, nonpolar, and that can even be separated down into the aromatic amino acids. I mentioned that amino acids are always levorotatory. They are all S except for cysteine. We talked quite a bit about the acids and bases of amino acids, the branched chain and the beta branched. We talked about disulfide bonds. We talked about how the aromatic nature of these side chains affect how it interacts with light. We went over the various rules for that. I talked about the dihedral angles and the peptide bond formation. And I talked about the various modifications. So I think that was everything I wanted to talk about in this amino acid video. In the next video, I will talk a bit more about proteins, so the polypeptides, the polymers of amino acids making up the proteins, and so we will see how these amino acids come together and form the workhorses of our biochemistry. And I will see you in that video.